So before we understand what exactly is an in vitro packaging, again we will quickly go through the late transcription and DNA packaging. The most important one is going to be the different stages what we have already studied. Alright, the early event is going to be the synthesis of head and tail proteins. All these head and tail proteins are all present in the freely soluble form. Alright, so the next stage they could identify is going to be the scaffolded pre-head. But in the scaffolded pre-head, again DNA packaging is not possible, basically because several protein subunits has to be removed. That removal is done by the gene product GrowE. Alright, so then several scaffolds or unwanted protein structures have been eliminated. Now that results in the formation of what is known as a pre-head. Alright, so now when it comes to the pre-head, the next stage is going to be the assembly of the concomitant DNA repeats of lambda DNA, that is during the DNA replication. So now two cause sites with the help of NU1 and A will come very close to the mouth of the pre-head region. Then it is going to be packaged inside. Then once it is going to be packaged inside, then the capsid formation takes place. During this particular process, the D proteins are going to be assembled on the head to give a definite shape. Then because of the endonuclease activity, these things are cleaved. So now we have a head in which DNA is going to be encapsulated. Then separately the tail proteins are going to be assembled and they do get joined together. Now this is the various stages and what all the things is going to happen in the late transcription and DNA packaging. Once the number of copies is going to be increased, then because of the lysis due to R and S, so the cell is going to be lysed. So once the cell is going to be lysed, that became a transparent, and that we are going to observe as a plaque formation. So now in case of an in vitro packaging, in this particular life cycle, so we have identified several stages. Alright, now what are those stages? Okay, now the stage 1 is going to be the formation of a scaffolded pre-head. This is stage 1. And what is stage 2? This is going to be the pre-head, is the stage 2. Now stage 3 is going to be the DNA packaging. It will bind and then it will going to get DNA packaged. Okay, now this is the DNA packaging. Then the fourth stage is going to be the capsid formation. So for the capsid formation, what is required is the assembly of the D protein on the surface. This is very, very critical. Finally is the assembly of head and tail. Now this is the capsid. Then to this particular one, tail, which is independently assembled, will join together to form a viable lambda DNA particle. Alright. So now here you have to understand that, for example, so now we have several processes going to take place, isn't it? Suppose if there is a mutation in this D, what happens if there is a D? So then what happens? Further events is not going to take place. So now suppose if there is a mutation in the D gene, so whatever the lytic process takes place will stop at the pre-head region itself. Okay. Then another one, for example, if there is a mutation in the gene E, then what happens? We don't even get the scaffolded pre-head. We are all going to present all those gene product in the freely soluble form. So in case of an in vitro packaging, what we do that we are going to mix a lysate from one with a mutation of a D, another one the lysate with a mutation of E. Alright. Now, for example, we will take so the mutation, the strain is what is called as a BHB2690. This is a strain number. Alright. If you do take this particular one, if I try to do the lambda DNA and try to get into the lytic cycle, so now since there is a mutation in the D, we don't get viable particle, but most of the times what we end up is the pre-head itself. So we are going to have the pre-head accumulated. So suppose in this particular strain, if I try to take this strain and infect with lambda DNA, suppose then if I do take the lysate is rich only with respect to the pre-head. And this cannot be converted into a viable product basically because the D protein is absent. Suppose to this particular one, if you add D protein, then this pre-head will be converted into a viable particle. Then how do you get the D protein? Now the D protein we are going to get it within another lysate in which the mutation is with the gene E. So now here in case of this mutation E, what happens? We don't get the scaffolded pre-head. All the proteins are going to be translated and are present in the soluble form, including D protein. So, okay. So now in this mutation, we have all the gene products translated into a soluble protein, including D. Now what we do in case of an in vitro packaging is that we take the lysate from this mutation, lysate from this mutation. If you add together, now this particular one has got the pre-head but do not have the D. So here we have everything but not the pre-head. So now the D protein is complemented from this lysate. So when you mix these two things with our cloned DNA, so now we have the left arm, then we have the DNA which is inserted, then we have the right arm with the cos site. Then what happens? This particular DNA, since they do have the pre-head, so it is going to, because it has got two cos site, so it will bind here, it will going to get packaged here, and then because it lacks D, it never completed. So now this particular strain or the lysate will provide D. Then what happens? So it is going to be a viable particle. So now what happens is that, so now we have taken our clone and we make sure that it is packaged into the head region and followed by this. So now if you do take this particular one, this is a viable particle. Now with this, if I just take the E. coli host, so now this particular one has got the enzyme so that it will lysate this, the cell membrane, and then it will insert easily into the host cell. So the number of plaque we are going to get is again 1 into 10 to the power of 10 plaques. So now this is the strategy what has been employed. So with respect to the phage vector, so in order to have the phage vector and then we have got the clone, and for the transfection it has to be with in vitro packaging. And how the in vitro packaging is going to be done, so for that this lytic cycle understanding is very important, and the mutation which takes place in E strain is very important. In case of the mutation D, the lysate only contains the pre-head, whereas in case of another lysate with mutation E, we only get the soluble proteins including the D protein. When you mix these two lysate along with our cloned DNA, so now that is going to get packaged and we are going to get a viable particle. Now you take this particular in vitro packaged DNA particle, so now you take the host, then if you do an infection, now we are going to get a good number of plaques so that we can study for further analysis, isn't it? So this is exactly what is called as an in vitro packaging. So again I will repeat. So now this is the left arm and this is the right arm. Suppose if there is no insertion, then only the left arm and the right arm again they join together. And now if you just calculate what is the DNA size, for sure this DNA size is less than 36.4 kilobase. So now this is not going to get packaged. So already we have a selection procedure. So now if I do take the head and tail region, and this is the lytic cycle, and we have removed the lysogeny cycle. We have inserted the DNA of our choice, something around like 24.6 kilobases, and here we do have the 2 cos site. Now with the help of the in vitro packaging here, so we have the lysate derived from mutation D, which only contains the pre-head, as well as the mutation E, which contains only the soluble D protein. It is going to get packaged. All right. Now during the in vitro packaging, they have made 2 critical observations. What they have found out that in case of an in vitro packaging, the most important one is the 2 cos site, and next one is going to be the distance between these 2 cos site. Doesn't really matter what is the DNA content from here to here. Okay, I will repeat one more time. So in case of an in vitro packaging, the lysate or the whole the mixture, the enzymatic reactions takes place. So now here the critical one is going to be the 2 cos site. Second one is going to be the distance between 2 cos sites. I told you there is a minimum and a maximum. 36.4 kilobase, anything less than that, it is not going to get packaged, and anything above 48.5, again it is not going to get packaged. So that distance is very important. So now based on this observation, since these ends are important, not the DNA whatever is going to be present here, they thought, why can't we construct a cosmid vectors? Cosmid vectors are nothing but plasmids with cos sites. They are called as a cosmid vectors. So now in case of a Forge vectors, we can add the DNA up to 24.4 kilobases. Alright, so 24.6 kilobases. So now in case of a cosmid vectors, all it requires is 2 cos site, and we are not, because since we already have the head and tail pre-made, now this DNA region is also not important, and already it is in the final stages of the lytic. This one is also not important. So as a result, if we do have a 2 cos site, so now we can, from here to here, the entire thing can be a foreign DNA. So compared to Forge vectors, cosmid vector is highly evolved, in which if it is 24.6 kilobase is the limit in case of a Forge vector. In case of a cosmid vector, we have 38 to 50 kilobase can be easily inserted in the cosmid vector. So because of this particular advantage, rather than the Forge vectors, cosmid vectors are very, very important. And what are cosmid vectors? It is nothing but the plasmid vectors. Now I told you that the most of the plasmid vectors do contain origin of replication, antibiotic resistant gene, polyclonal site, so many things. So now in case of a cosmid vector, again this is the same plasmid but also contains cos site, isn't it? So now with cos site, how are we going to do the cloning, and then how are you going to do the transfection? We will study in the coming videos. But in case of a Forge vector as well as in case of a cosmid vectors, critical step is going to be the in vitro packaging. So once we have understood what exactly is a in vitro packaging, then rest of the cloning stuff is going to be very, very easy. Now here I have given some of the names of the cosmid vectors. This is MUA3. Now what I have written is the size of this particular cosmid vector is 4.76 kilobase. This can accommodate the DNA length from 40 to 48 kilobase. Now PJB8, this is 5.4, it can accommodate 32 to 45 kilobase. Then we have the Homer 1 series, 5.4 kilobase with 30 to 47 kilobase. Homer 2, 6.38 kilobase and 32 to 44 kilobases. And this is PJC79, 6.4 kilobases, then it can accommodate from 32 to 44. Now you have understood that we started with plasmid vectors, and we know that in the plasmid vectors we can only insert maximum 6 kilobases. So we started looking for a newer vector Then we look into the forge vectors. We studied the complete life cycle, lytic, lysogeny, the arrangement of genes. So then we designed a forge vectors. In that we have both insertional vectors as well as replacement vectors. We took the help of beta galactosidase, that is the LAG-Z DNA, C1 repressor gene, red and GAM gene. So now we have formulated that one. So for the efficient transfections, so we have designed an in vitro packaging consisting of mutated lysate. One lysate will have a mutation in the D, which contains only the pre-head. Another lysate which has got a mutation in the protease E, which only contains soluble proteins. When you combine these two lysates along with our clone, they are going to get packaged into the head region. So we are going to have a viable particle. Now this viable particle, again we infect that into E. coli so that we are going to get the plaques of our interest. Alright. Now during this particular time, again we try to understand the events that normally takes place in case of an in vitro packaging. They realize that in case of an in vitro packaging, already we have a pre-head and already we have soluble proteins and everything. All it matters is to cause site. Doesn't really matter whether you have the head and tail DNA region or a lytic DNA region. Okay. The next one is going to be the distance. If it is too small a distance, then it is not going to get packaged. Larger the DNA segment, not going to get packaged. So if this distance is maintained, in which we have 36.4 to 48.5 cases, that is automatically get packaged. So because of that, all right, why can't we design another vectors? So it is nothing but a plasmid vectors. They have taken it and they have inserted cos region into this plasmid vectors. That automatically is going to be the cosmid vectors. Now using these cosmid vectors, what are the strategies we have involved in the cloning procedure? We will discuss that in the next video.